Hello guys, this is Jyotiswar. Welcome to my channel. Today in this video we are going to discuss about bio chapter 4, that is absorption of roots, the process involved. So in our previous video we have completed detailed osmotic pressure. Today we will learn about tonicity, active transport, turgidity and felicity. Okay, so first, what do you understand or what do you mean by tonicity? So here tonicity means it is a relative concentration of the solution that determines the direct and extent of diffusion. Okay, so based on its solution they are classified into three types. One is isotonic, hypotonic and hypertonic. So first we will learn about iso. So what are those three? First I will let me explain. Iso, hypo and hyper. Okay, so these are the three. So first we will learn about isotonic. So what do you mean by isotonic? It is the relative concentration of water molecules and the solute on either sides of the cell membrane in the same. In such a solution there is no network moment of water molecules across the cell membrane. That means no osmosis. Now coming to the hypotonic. So this means that the solution outside the cell has a lower solute concentration than the fluids inside the cells. As a result, the water molecules from the outside will move into the cells, which means endoosmosis. Now moving to hypertonic. So here in this condition the solution outside the cell has a higher solvent concentration than the fluids inside the cell. Consequently, the water molecules from the interior of the cell will move out. That is called exoosmosis. Okay. So for this, what will be the result? Okay, so first coming to the isotonic, the result will be that cell shape and size remains unchanged. When coming to hypertonic, cell shrinks in size and loses shape. When coming to hypertonic, here cell slightly enlarges or even bursts also in some cases. Okay, that is about tonicity. Now moving to active transport. Okay, so here what do you mean by active transport? Okay, so active transport means that it is a passage of a substance from its lower to its higher concentration through a living cell membrane using energy from the cell. That is called active transport. Okay, and active transport in is in a direction opposite of diffusion. In our previous video we have already learned about diffusion. So the passage of active transport is opposite of what diffusion. Okay, so that is about active transport. So now certain nutrients such as ions of nitrates, phosphate, potassium, zinc, manganese, etc. cannot pass through the cell membrane of the roots easily. So this is because the concentration is higher inside the root cells, and it is so maintained in order to develop osmotic pressure for absorbing water. Okay, that is about active transport. Okay, so now that is about active transport. Now we will understand about passive transport. Okay, we will understand about passive transport. So here what do you mean by passive transport? Passive transport is nothing different from diffusion. Okay, passive. Okay, the word passive refers to the requiring no input of energy. There is a free movement of molecules from higher concentration to their lower concentration. That is passive transport. Now moving to turgidity and flaccidity. So here, now what do you mean by turgidity? Here, turgidity refers when a cell reaches a state where it cannot accommodate any more water that is, it is fully distended or fully filled, it is called turgid, and this condition is called turgidity. Okay, that is turgidity. Now when a cell in turgid, its walls is stretched under pressure from inside, and in its turn it presses the cell contents towards the cell of the center of the cell. So the pressure of the cell contains on the cell wall is called turgor pressure, and pressure exerted by the cell wall on the cell content is called wall pressure. Okay, that is about turgidity. Now I will give a simple and easy definition for turgidity, felicity, and also plasmolysis. Okay, so first turgidity. So now I will explain about turgidity, and this will be a simple definition. Okay, it may be a term. Okay, so now turgidity is a state of a cell in which the cell wall is ridged and stretched by an increase in volume of vacuoles due to absorption of water. Okay, we have already discussed about that. Now the cell is then said to be turgid. Okay, turgidity is nothing but when it is turgid, this condition is called turgidity. Okay, that is turgidity. Now moving to felicity. So here felicity is a condition in which the cell content and the cell is no more tight. Okay, the cell is then said to be felicid. Okay, now moving to plasmolysis. So here this is the contraction of cytoplasm from the cell wall caused due to withdrawal of water when placed in a strong solution. It may be a hypotonic. That is about plasmolysis. Okay. Okay, now we will see about the uses of turgidity to plants. What is the use of turgidity? What is the use of turgid? What is the use of this turgidity in plants? So the first one is turgidity provides a rigidity to soft tissues such as leaves, and the second one is turgor pressure helps to push through the hard grounds, and the third point is turgor in root cells build up root pressure, and also turgor in the opening and closing of stomata, turgor movement. Okay, these are the useful things. Okay, useful turgidity for plants. Okay, so that's it guys, we will continue in our next video. Till then, bye bye, but don't forget to like, share and subscribe.